good afternoon, students. So welcome you all to another session, right? So in the previous session, we have started discussing a few things with regards to the types of immune responses, right? Pertaining to something called as the acquired immunity, right? So with regards to the acquired immunity, we started taking up with the types of acquired immune system based on the type of immune response that is being elicited by our system. So in connection to that, we spoke about two different types of responses. One, we call it as cell-mediated immunity, and the other one is called the humoral immunity, right? So in the previous session, we began with the cell-mediated immunity, and we saw in brief how exactly is the cell-mediated immunity actually working out. Right, so we saw there was something called as the T lymphocytes, and then we saw about something called as the antigen presenting cells, the APCs. Right, so how the antigen presenting cells would process the antigen and how they would present the antigens to the T lymphocytes. So that is what we had actually discussed in the previous session, right? So I hope all are got a clear picture about how the cell mediated immunity would actually work. So we also saw that when does the cell mediated immunity come into picture? Whenever there is some sort of viral infection in our body, and at the same time, whenever there is some sort of cancerous cell proliferation in our body, and along with that, during the process of graft rejection, I told you that this cell mediated immunity would play a very, very important role. Right. So that's what we have discussed in the previous session with regards to the cell mediated immunity. Right. So in this session, right, in today's session, let us take up the second type of immune response that is being evoked, right, which is called as the humoral immunity, right? So let's begin with the humoral immunity. Right. So let's begin with this humoral immunity. Now, what does this humoral immunity mean? I just gave you a brief description in our previous sessions when we actually began with the classification, right? Initially, when we started off with cell-mediated immunity as well as the humoral immunity, I just gave you a brief background of what do we mean by humoral immunity, right? So what exactly is this humoral immunity? I told you all to remember that humoral immunity is nothing but B cell-mediated immunity, right? Whereas in case of cell-mediated immunity, it was majorly the T lymphocytes. Now, in case of humoral immunity, it would be majorly the B lymphocytes who is actually involved in this particular process. Right. Now, why do we call it as humoral immunity? This also I have already made a mention of this. Why do we call it as humoral immunity? Because the antigens, when they enter inside our body, we know that the B lymphocytes would proliferate, it would differentiate to form all that process we saw, and finally there is formation of antibodies. Once the antibodies are actually formed, those antibodies are actually released into the body fluids. In the body fluid, right, in the body fluids, the antibodies would combine or would bind to the antigens in a specific lock and key mechanism. Right. Since the interaction between the antigens and antibodies is usually taking place in the body fluids, which we also call it as humor, here we start referring to this as the humoral immunity. Right. So humoral immunity, the immunity which is actually developed in the humor, that is nothing but the body fluids that are present in our body. Right. So the binding of antigen and antibody, right, or the interactions between the antigen as well as antibody, usually they take place in the humor, that is the body fluid. Therefore, we start calling it as the humoral immunity. So let's get into the mechanism of humoral immunity. What exactly happens in this humoral immunity? We have already seen this, right? But just a quick recap of what we have already seen with regards to the cell-mediated immunity. We spoke about all of this mechanism when we were talking about the B cells itself, right? There I told you how exactly the antibodies are produced. The same type of mechanism would be followed, or it would be applied here also, right? So let's begin with the mechanism of humoral immunity. Again, let us continue with the same. Just imagine that there is a pathogen entering inside our body, right? So here we have this pathogen being entering inside our body. This would be the antigen, right? So this is the antigen which has entered inside the body. Now, once the antigens enter inside the body, here we have this B cell that is being present, right? So this would be our B cell. And I've also told you that on the surface of the B cell, there are certain receptors which are present. We call it as B cell receptors. right? And also when we were discussing about the types of antibodies, we spoke about two different classes of antibodies, right? One was called the IgD as well as IgM. So those we said that they are actually considered as the membrane bound antibodies, right? So the antibodies which are bound on the surface of the B cell. Right. Now, whenever the antigen enters into the body, the receptors which are present on the surface of the B cell will start recognizing that antigen. Right? It would start recognizing this antigen. It would come and bind to the receptors that are present on the B lymphocytes or the B cell. Once they bind to this B lymphocyte, we are actually talking about a matured B lymphocyte. We know that the B lymphocytes are formed in the bone marrow. Maturation also occurs in the bone marrow. After maturation, they will have these receptors present on their surface. Right. So these receptors would start recognizing the antigen that has entered into the body. Once they recognize the antigen, they would bind to the antigen, and binding of antigen to the receptor on the B cell is the signal that is given for the B cell. So what signal is actually given to this B cell? Signals are given to the B cells such that this B lymphocyte would now start undergoing the process of multiplication, or rather they would undergo the process of activation. Right. Upon activation, we have already seen that the B lymphocytes would get differentiated into two types of cells. Right. So upon activation, the B lymphocytes would now get differentiated into two types of cells. So we know what are those two types of cells. One is called the memory cells, and the other one is called as the plasma cell, or we also call it as the effector cells, right? So it would differentiate into two types of cells, memory cells as well as the plasma cells or the effector cells. Now this effector cells, based on the type of antigen that has entered into the body, the effector cells or the plasma cells will now start synthesizing the antibodies. Right? So they would start synthesizing these antibodies. Now again, remember, all of these antibodies are said to be specific to the specific antigen that has entered into the body. Right? So all the antibodies which are being synthesized by this effector cells or the plasma cells, they are said to be highly specific to the particular antigen that has entered inside our body. Right? Now, these antibodies which are being synthesized will now go and start targeting this particular antigens that has entered into the body. Now, before that, once these antibodies are formed, these antibodies are actually now released into the body fluids. They are released into the body circulation. In the body fluids or in the circulation, right? Or we can put it this way. In the body fluids, these antibodies will go and combine with that of the antigen. Right? It will go and combine with that of the antigen. Right? So once they bind to this antigen, we have the antigen antibody complex that is being formed. So this antigen antibody complex is nothing but a lock and key mechanism, what we have already discussed, right? So the antigens would bind to the antibody in a lock and key mechanism. That's the reason why we say each antibody is said to be highly specific to a particular antigen that has entered inside the body. So these antibodies, which are synthesized from the effector cells or the plasma cell, would now enter into the body fluids, right? In the body fluids, they would go and bind to this antigen in a lock and key mechanism. Once they are bound to the antigen, we start calling this interaction as antigen-antibody interaction. And because of this interaction, the antigen would be completely destroyed. Right? So there are different ways how the antibodies will encounter, or we can put it this way. There are several ways how the antibodies would target the antigen. So we'll see what are those different ways in a few moments from now, how exactly the antibodies work in bringing about the destruction of this antibody. Right. So this entire process we start calling it as humoral immunity. Right. So we call it as B cell mediated immunity. Why do we call it as B cell mediated immunity? Because it is the B cell alone which is actually involved in this process of immune response. Whereas in case of cell mediated immunity, we saw that there were two types of cells, right? One was called the antigen presenting cells and the other one was T lymphocytes. So both of these cells together, right, in a coordinated fashion, in a coordinated manner, they would bring about the destruction of the antigens that has entered into the body. That we started calling it as the cell-mediated immunity. And here we are talking about humoral immunity because the antigen-antibody interaction usually occurs in the body fluids, that is humors. Therefore, we also start calling it as the humoral immune response, or simply we can call it as humoral immunity. Right? So this is what is exactly happening. Right? So quickly going through it once again. As and when the antigens enter into the body, right, the B cells will start recognizing this based on the receptors that they have on that surface of the mature B lymphocytes. Right. Once the antigen binds to the receptor present on the surface of the B lymphocyte, this particular B lymphocyte will now start undergoing the process of activation. Right, it will start undergoing the process of activation. Upon activation, this B lymphocytes would get differentiated into two types of cells: memory cells and the plasma cells. Right. The plasma cells will now, based on the type of antigen that has entered into the body, the plasma cells will now start synthesizing or secreting the antibodies, specific antibodies against the specific antigen that has entered inside the body. Right. So once the antibodies are being synthesized, these antibodies are released into the body fluids. In the body fluids, the antibodies will go and bind to the antigen, right, forming an antigen-antibody complex, right? And this interaction is a lock and key type of interaction. Therefore, it is said to be highly specific. Now, since this interaction is formed in the body fluids, or it is usually taking place in the body fluids or the humors, we start calling it as the humoral immunity or humoral immune response. Right? So this is what we have. Now, what will the memory cells do? These memory cells will now start remembering this antigen that has entered inside the body, right? Such that second time, whenever the same antigen enters into the body, at that point of time, this process will not occur. Directly, this memory cells will go and start targeting this antibody. So at that point of time, there is no differentiation required. Differentiation of B cells is not required. Memory cells itself will directly identify the pathogen that has entered into the body or the antigen that has entered into the body, and it would bring about the destruction of that particular antigen inside the body fluids. So this type of immune response, we start calling it as the humoral immunity. Right? So this is what we have with humoral immunity. So two types of immune responses we have seen so far. One is the cell-mediated immunity. Second one is this humoral immunity. Right? So based on the type of immune response showed by the acquired immune system, we start classifying acquired immunity into two types. One is called the cell-mediated immunity, and the other one is humoral immunity. Right. Now, how does the antibody target the antigen? So what are the different ways in which the antibody targets the antigen? So let's take up with that. So different ways in which the antibody targeting the antigen. So let's take up with that in brief. Right. So what we are taking up is action of antibody against the antigen. So how does the antibody target the antigens? Right. Number one, what the antibodies does is, antibodies, once they are actually formed and once they are released into the body fluid, they would bind to the antigen and bring about the neutralization of the antigen. So the first way how the antibodies act is, they bring about neutralization, or they neutralize the antigen that has entered inside the body. We call it as neutralization reaction. Right. So they bring about neutralization of the antigen that has entered into the body. This is one way how the antibodies would target the specific antigen that has entered inside the body. What do? They start neutralizing the antigens that has entered inside the body. First one. The second way what they do is, right? Second way how the antibodies act is, the antibodies would bring about a process called as agglutination. Right? They would bring about a process called as agglutination. Agglutination of insoluble antigens. Right? Agglutination of insoluble antigen. If the type of antigen that has entered inside our body is an insoluble antigen, right? If it is an insoluble antigen, it would bring about the process called as agglutination. So agglutination is nothing but clumping, clotting. right? It clots or it brings about clotting or clumping of that antigen such that that particular antigen is no longer able to multiply, it is no longer able to proliferate, or it is no longer able to show its activities, or it is not able to move from one place to the other place. So all the antibodies would come and bind to this particular antigen, bringing about the process of agglutination. That is the second way how the antibodies would target the antigen. Number one, as and when the antigens enter into the body, one way how the antibodies act is they start neutralizing the antigen. They bring about something called as the neutralization reaction. They start neutralizing the antigen. Second way how they act is they bring about agglutination of this particular antigen. If the type of antigen entering into the body is an insoluble antigen, at that point of time, they would bring about agglutination or clumping or clotting of this particular antigen. The third way how they act is they bring about precipitation, right? So they bring about precipitation of soluble antigens, right? So they bring about precipitation of soluble antigen, right? So if the type of antigen entering inside the body is insoluble antigen, at that point of time, they would bring about the process called as agglutination. Whereas if the type of antigen entering the body is a soluble antigen, at that point of time, they would bring about precipitation of that particular antigen. Right. So three ways we have seen how the antibodies would target the antigen. Number one, they bring about neutralizing the antigen that has entered inside the body. So they neutralize the antigen. Number two, they, if at all there is an insoluble antigen entering inside our body, that insoluble antigen is undergoing a process called as agglutination. So these antibodies will start agglutinating or clumping or clotting this insoluble antigens that has entered inside the body. Number three, if the type of antigen entering into our body is a soluble antigen, at that point of time, what do? They bring about the process called as precipitation. So I hope all are familiar with precipitation, so we are not getting into much details of it. So three ways. They neutralize the antigen, they bring about agglutination of insoluble antigen, they bring about precipitation of soluble antigen. Right. So these are the three ways. Next one. Number four. How do they act? Just suppose there is some sort of bacteria entering into the body. Just suppose that there is a bacteria entering into the body. How do the antibodies target the bacteria? They would now bind to the flagella of the bacteria. Right. So these antibodies, once they are synthesized or released, would bind to the flagella of the bacteria. Now we know what happens if the antibodies bind to the flagella of the bacteria. So we know what is the role of flagella also. Flagella is mostly responsible for the process of locomotion. Now if these antibodies go and bind to the flagella of the bacterium, the bacterial cell will no longer be able to move. So if they are no longer able to move, what will these antibodies do now? They bring about immobilization, right? Immobilization, right? They bring about immobilization of bacteria. So how are they bringing about this process of immobilization of bacteria? By binding to the flagella of the bacteria. So once they are bound to the flagella of the bacterium, the bacterial cell will no longer be able to move. As a result of which, it would remain in that particular place itself. Right. So four ways we have seen how the antibodies would start targeting the antigen. Number one, they would neutralize the antigen. Number two, they would bring about agglutination of the insoluble antigen. Number three, they would bring about precipitation of the soluble antigen. And number four, they would bring about immobilization of the bacteria. How do they bring about immobilization of the bacteria? By binding to the bacterial flagella. As a result of which, the bacterial cell will no longer be able to move from one place to the other place. Four ways we have seen. Right. Now, what do they do? After the antibodies have bound to the bacteria, they neutralize it, they agglutinate it, they precipitate it, and they immobilize the bacteria. Once all of these processes are done, right? Once all of these processes are done, the next process what they do is, right? So the next process what they do is they go and start activating the phagocytic cells. You can put it this way. They bring about activation of phagocytic cells. They bring about activation of phagocytic cells. right? So they start bringing about the process of activation of the phagocytic cell. Once the antigen is being bound by the antibody, either through the neutralization reaction or through agglutination reaction or through precipitation reaction or even by immobilizing the bacteria also. Now what do? Some of the antibodies will start sending signals to the phagocytic cells. So what will the phagocytic cells do? We are very much clear with phagocytosis, right? So what does the phagocytic cells do? The phagocytic cells will now arrive to that particular point where the antibodies have been bound to the antigen, and they would bring about the process of phagocytosis of that particular antigen. Either it be through neutralization, agglutination, precipitation, or immobilized bacteria, right? So any of the pathogen, right? So they now would arrive to that particular location and they would bring about the process of phagocytosis, right? So once phagocytosis is done, all the phagocytic particles or fragments are being formed. Yesterday we saw that, right? After phagocytosis, what would happen? Phagocytosis by macrophages, antigenic fragments are being released. Those antigenic fragments are now, they bind to the MHC molecules, either class 1 or class 2 molecules. So processed antigens on class 1 and class 2 MHC molecules would now be taken to the T lymphocytes. They would present the antigen to the T lymphocyte, and the T lymphocytes would now bring about complete destruction of this particular antigens, right? So they bring about the process of activation of phagocytic cells. Now, once this process, right, this process of activation of phagocytic cells, we also call it as a process called as opsonization. Right, so we also call it as opsonization. So what exactly is this process called as opsonization? Opsonization is nothing but the process of phagocytosis, which is mediated by the antibodies. Right, so let me repeat it. What is phagocyte? What is opsonization? The process of phagocytosis, which is mediated by antibodies. Right, so the antibodies which mediate phagocytosis, we start calling them as opsonins. Right, opsonins are nothing but substances which mediate substances which mediate the process of phagocytosis. We start calling them as opsonins. So here in this case, antibodies are usually referred to as opsonins because antibodies are the ones which go upon binding to the antigens. Antibodies are the ones which go and bring about activation of the phagocytic cells such that they undergo this process called as opsonization, right? So now what is opsonization? Opsonization in simple, if you want to remember opsonization, you can remember it this way. Antibody mediated phagocytosis, we start calling it as opsonization. Antibody is usually referred to as opsonin because antibodies are the ones which are actually mediating phagocytosis, right? So we have other substances also which mediate the process of phagocytosis, but now we are not worried much about it, right? Let us get to this particular point, right? So how does this antibodies usually work in targeting the antigens that has entered into the body? Number one, as and when the antigens enter into the body, they either undergo neutralization reaction. So through the neutralization reaction, what the antibodies are doing is they are locking up the antigen so that the antigen will not be able to escape from the antibodies. They neutralize it. Second, if it is an insoluble antigen entering into the body, they bring about agglutination of that particular antigen. Clumping. Again, what they are doing? They are locking the antigens so that the antigens would not escape. Number three, if it is a soluble antigen that has entered inside the body, this soluble antigen, they undergo the process of precipitation. Again, by precipitating the antigen, they are locking the antigen so that the antigen would not escape. If there is some sort of bacterial cells entering into the body, these antibodies would bind to the flagella of the bacterium such that they start immobilizing the bacteria. Again, through this immobilization process, what exactly they are doing? They are locking the bacteria so that the bacteria will not escape. Now, once all the antigens are being locked, either through neutralization reaction or agglutination reaction or precipitation or through immobilization, once the antigens are being locked, then the antibodies would start sending signals to the phagocytic cells. They would start activating the phagocytic cells to undergo the process called as opsonization. right? So since antibodies are involved in activating the phagocytic cells, antibodies here are referred to as opsonins, right? So opsonins mediated phagocytosis or antibody mediated phagocytosis. In this case, antibody mediated phagocytosis, we start calling it as opsonization, right? So through opsonization, phagocytosis is done. Through phagocytosis, whatever antigens here have been locked are being completely destroyed. I mean, they are being fragmented with regards to what we have seen yesterday. Processing of this antigen is done. Once antigen processing is done, antigen presentation would be done to the T cells. After antigen presentation to the T cells would now bring about complete destruction of this antigen. Right. So these are some of the ways how the antibodies will start targeting the antigen. One more way how these antibodies target the antigens is they bring about by a process called as activation of, right? So they bring about the activation of something called as the complement system, right? So they bring about the process called as activation of this complement system. Activation of complement system. Now, what is this complement system all about? Complement system is another one more important immune system present in our body, right? It actually falls under the category of innate immunity, right? It usually falls under the category of innate immunity. More precisely speaking, it usually falls under the category of the second line of defense, right? A part of innate immune system, complement system, right? This is again one of the complex immune systems that is present in our body, right? So there are several proteins which we call them as complement proteins which are involved in this particular system. There are almost 11 to 12 different types of complement proteins which are involved. We call them as complement 1, C1, C2, C3, C4, like that we start naming them, right? So all of these complement proteins, they are involved in bringing about the destruction of some of the pathogens that have entered into the body, right? So there is a cascade of reactions happening. There is a series of reactions happening. So through this cascade of reaction or series of reaction, the complement system would be activated. Finally, once the complement system is activated, one of the last complex, we start calling it as membrane attack complex. In simple, we call it as MAC, right? Membrane attack complex. So this membrane attack complex, which is the last component of this complement system, it would bring about the destruction of any bacterial cell or the viral cell that has entered into the body, right? So whenever any bacterial cell or viral cell enters into the body, we have this complement system getting activated. So this complement system upon activation, right? So this complement system upon activation would now bring about complete destruction, right? Destruction, we can put it this way, it brings about the lysis, right? It would bring about the lysis of either the bacterial cell or the viral cell through this membrane attack complex, right? So they usually attack the membranes of the cell, either the bacterial cell or the viral cell. Once the membrane is being attacked, there is complete destruction of that particular pathogen that is entered into the body. That's the reason why we start calling this complex as the membrane attack complex, right? So this is what we have, 6 different ways how the antibodies function, or 6 different ways how the antibodies target the antigens once they have entered inside the body, right? So quickly going through it. First one, they bring about the process of neutralization, right? They bring about the process of neutralization of the antigen, right? So once they neutralize the antigen, what actually they are doing, they are locking the antigen so that the antigen would not escape. Second one, if there is an insoluble antigen entering into the body, that insoluble antigen, they start agglutinating the insoluble antigen. They start clumping, they start clotting the insoluble antigen that has entered into the body. Again, if there is a soluble antigen that has entered into the body, they bring about the process called as precipitation. So they start precipitating the soluble antigen that has entered inside the body. Then, if at all there is any bacterial cell entering into the body, those bacterial cells are immobilized. So how are they immobilized? These antibodies would now go and bind to the flagella of the bacterium. So once they bind to the flagella of the bacterium, the bacterial cell will no longer be able to move. Therefore, they are said to be immobilized. So through all of these 4 processes, what the antibodies are doing is they are locking the antigens, right? So they are locking the antigens such that the antigens would not escape. So once they have locked the antigens, these some of the antibodies would now go and start activating the phagocytic cells. Similarly, some of the antibodies would go and start activating the complement system. Right. So through the process of activation of phagocytic cells, what exactly is happening? There is a process called as opsonization that is happening. Right. So opsonization, as I've already said, antibody-mediated phagocytosis. In this case, we can consider it that. But actually speaking, it is opsonal-mediated opsonization. Right. So there is this opsonal-mediated opsonization happening. Right. So here, why do we call it as antibody mediated? Because the antibodies are actually involved in this particular process of activation of the phagocytic cells. Right. So they bring about the process of opsonization. That is, all of this particular antibodies, they start targeting the antigen. Once they target the antigen, they go and activate the phagocytic cells. Once they activate the phagocytic cells, what is happening? These phagocytic cells would now bring about the process of phagocytosis. That process we start calling it as opsonization, and the antibodies here we refer to them as opsonals. On the other side, along with the activation of the phagocytic cell, they go and start activating the complement system also. So upon activation of the complement system, whatever bacterial cell or the viral cell which has entered into the body, right? So this complement system will start targeting the membranes of that particular system. As I've already mentioned, it is a complex system involving almost different types of complement proteins, right? Different types of complement protein. The last component is this membrane attack complex. So it is a cascade of reaction. Through this cascade of reaction, once the membrane attack complex gets activated, this membrane attack complex will now start targeting the membranes of the bacterial cell as well as the viral cell. It would bring about the complete lysis of the bacteria as well as the virus. Right? So this is how the antibodies act upon the antigens that has entered inside the body. Right? So this we have with this humoral immunity. Right? So with this, we are done with our topic called as the cell-mediated immunity as well as the humoral immune response. So cell-mediated immunity, it is mostly the T-cell-mediated immunity. Along with that, we also have something called as the antigen-presenting cells, which are involved in bringing about the immune response. Second one, we have humoral immunity. Here we usually call it as the B-cell-mediated immunity because it is completely related to the antibodies, right? So the antibodies, once they are released, they move into the body fluids. In the body fluids, they would bind with the antigen-antibody interaction. Once the antigen-antibody interaction is done, since this sort of interaction takes place in the humus or the body fluids, we start calling it as humoral immunity. So what sort of interaction? It is a lock-and-key type of interaction between the antigen as well as the antibody. So how does this antibody bring about targeting the antigens? These 6 different ways. Neutralization, agglutination, precipitation, immobilization, activation of the phagocytic cell, which we call it as opsonization, as well as the activation of the complement system. Right? So this is what we have with the classification of the acquired immune system based on the type of immune response that is being evoked. Right? So with this, we are done with our topic called as classification of the acquired immune system. Yeah, right? So cell-mediated immunity as well as the humoral immune response. Right? Okay. So once we are done with this, let's begin with another topic, right, which is called as vaccination. Right? So the next topic, what we'll be taking up, is called as vaccination. Right? So once we are done with humoral cell-mediated immunity and the humoral immunity, the next topic, what we'll be taking up, is called as vaccination. So the next topic, what we have in the list is something called as vaccination. So what exactly is this vaccination? So we have some sort of idea with regards to this vaccination. Now also, even in the current days also, we have been listening a lot with regards to the process called as vaccination. We are actually speaking, everyone is actually waiting for a vaccine. Vaccine for what? COVID-19. Right. So let's take up a few things with regards to the topic called as vaccination. Again, the topic called vaccination is a very vague topic. There is too much to study, but since we don't have so much time and we don't have so much in our syllabus also, right? Just the brief of vaccination, right? Just a brief of vaccination we'll take up. Okay. So before we begin, what do we mean by the term vaccination, right? So what do we mean by the term vaccination, right? So vaccination is the process of immunization, right? The process of immunization, right? So what is vaccination? Vaccination is the process of immunization. Immunization against what? We are getting immunized against a particular pathogen, right? It is immunization against a particular pathogen, right? So what is this vaccination all about? Vaccination is the process of immunization against a particular pathogen, right? So what do we mean by this? Getting immunized against a particular pathogen, right? So we are actually preparing ourselves to fight against a specific pathogen in the future, right? So we are actually preparing our immune system to fight against a particular pathogen in the future or the near future, right? So that process is called as vaccination, right? So the process of immunization, right? The process of immunization against a particular pathogen. Now, how do we get immunized against a particular pathogen? How do we get immunized against a particular pathogen? How do we get immunized? By artificial inoculation, right? By artificial inoculation. I hope all are familiar with the term inoculation, so we are not going to get into much detail of it. By artificial inoculation, right? By artificial inoculation of what? Artificial inoculation of either killed pathogen, right? Either killed pathogen or attenuated pathogen, right? By artificial inoculation of either the killed pathogen, right? So we are killing the pathogen and we are injected into the system, or attenuated pathogen. Attenuated is nothing but weakened pathogen, right? So we are weakening the pathogen and we are injecting into a healthy individual. So this is how we actually define the term called as vaccination. So how do we define vaccination now? Vaccination is a process of immunization against a particular pathogen. So how do we go and by it? By artificial inoculation, right? By artificial inoculation of killed pathogen or attenuated pathogen into a healthy individual. So what exactly we are doing here? What do we actually mean by this particular definition? Right. So we bring about the immune response in a particular healthy individual. How do we bring about, as I've already mentioned in the beginning of this session, right? When we started off with vaccination, what are we doing in vaccination? We are preparing our immune system, right? We are preparing our immune system to fight against a particular pathogen in the future. So how we need to prepare our immune system? How do we prepare our immune system? By inoculating, right? By artificial inoculation of either the killed pathogen or the weakened pathogen, right? So by administering or by taking in the killed pathogen, say suppose we want to prepare ourselves against, say, a particular disease called as typhoid or hepatitis. Let us take hepatitis, right? I hope all are familiar with hepatitis. So we want to prepare ourselves, we want to prepare our immune system to fight against hepatitis B virus, which we feel we may be getting it in the future. So for that, what we are actually doing, we are taking the hepatitis B virus, we are either killing the pathogen, killing the hepatitis B virus, or we are weakening the hepatitis B virus. So upon killing or weakening the hepatitis B virus, we are injecting it into our body. Now what happens once we inject it into our body? Our immune system will recognize that particular pathogen. But remember, our immune system does not know whether it is killed or attenuated or it is weakened. As usual, our immune system will start recognizing that pathogen as foreign, right? Hepatitis B virus as foreign, and the immune system will get activated. B cell activation, T cell activation, there is memory cells being synthesized, all that, and memory cells have recognized the hepatitis B virus now. Now, This time, whenever there is actual hepatitis B infection in our system, at that point of time, there is no differentiation required, there is no activation required. Directly, our memory cells will start recognizing the hepatitis B virus, and without the process of activation and differentiation, all that, our memory cells will directly bring about targeting that hepatitis B virus, protecting us from hepatitis infection. That is the essence of this process called as vaccination. So when we talk about vaccination, how do we define vaccination? It is a process of immunization against a particular pathogen by artificial inoculation of either killed or attenuated pathogen into a healthy individual. Right? So this is what we have it in brief, the introduction to the concept called as vaccination, right? So we'll stop here for today. In the next session, we'll get into some more details with regards to vaccination. What are the different types of vaccines available? How do these vaccines work, right? So all that we'll take up in the next session, right? Thank you.